I'm assuming we won't get started until 5 past. And there's quite a few of us here.
Yeah, I think UCL is not here yet. I didn't see them yet. If they are not coming, then I will take the minutes.
It's just me from UCL, but Matt not here yet.
Okay, yeah. And I also didn't see Dimitri yet.
Hi, everybody.
Hi.
Hello.
So are we successfully running this meeting from the Sydney Uni address?
Yes, the recording started.
Yes,
Oh, right.
recording started.
Sorry, I
Good
forgot
business.
that we should have waited until the start, but
No, that's great. That's great. So you now have master control.
yes.
Okay.
So we should be good on the UCED end now.
Right. That's magic. Okay. And so sorry to be slow to this. Are you chairing and UCL's taking notes?
Yeah, that's what it says on the agenda. I'm happy to keep
Magic.
do that.
Yeah, good.
Yeah, just with the recording, if I can't make it, then I'll just put a note in and you guys can use the UCL Zoom instead, because I don't think anyone else from UCED is likely to be showing up at the moment. Are we expecting anyone else today in particular?
Reckon we can get going.
Alrighty. Well, then I suppose we will get started. So just going off the agenda, I don't think it's been changed that much, but Series 1 Phenaramoles, current state of the SAR paper, etc. Matt?
Yeah, I had a look at it just an hour ago, and no, it's looking pretty good. So there are just a few comments left in the experimental bit, but not many. There's a bit of reference tidying and stuff. And there's a new section at the end that Hung has put in there about mechanism of action, which is great. There are some references there, some citation markers, numbers for papers that don't have anything in the list. So I think he's taken it from thesis writing and put it in there, which is fine. I definitely want to tweak that, but it would help if he can put in the references that he's linked to there so that we have those papers, because there are a couple that are obviously very relevant and we need to know what they are. So there's just an action on him is to add those references in.
Okay, I'll try and let him know that if it's in the minutes as well, that would be good.
Thanks. Yeah, great.
Okay, and then is Louisa here to tell us if she's been able to figure out these SCY molecules, been able to get them to Wendy? Don't think she is today, is she? No. And let's see what else. Ma, do you have any data on the Phenaramoles to share with us? I saw she was here. Yeah.
Yeah, actually, I didn't do MIC test recently. So Wendy, or Maria, will you have a discussion?
Yes, so I updated the molecule screens by Marij. I put them all in the list. I'm not sure if there were phenaramoles there, but I think they all have been screened. I also see that Melissa is here on the call. It's maybe also good to let Melissa introduce herself and why she's here.
Hello, everyone. Sorry, I'm on the road now. It's a pleasure to me to be here. I was invited by Wendy to enjoy this meeting with you. I do not work with mycetoma, but I'm from Brazil and I work with sporotrichosis here. It's a great public health problem. And then we were discussing about share some of the drugs to try to test here and see if we can find some drugs that can be used to mycetoma treatment and also to sporotrichosis to increase the spectrum of the drugs. So it's a great pleasure to be here again. Thanks, Wendy, and nice to meet you, everyone.
Thank you, Melissa. Maybe good for the others to know, because not everyone will be familiar with sporotrichosis. Well, mycetoma and sporotrichosis are both fungal skin NTDs. So what they have in common is that they are caused by fungi, but different fungi. And of course, all the drugs we develop have antifungal activity, and it would be good to test if they are active against the other skin entities too, because they are both subcutaneous infectious diseases. And of course, if they have a bigger spectrum, that might help in the end, if we in the end think of the clinical progression and in clinical trials. So that was my idea, maybe to expand the spectrum of mycetos a little bit to include the other fungal skin entities too. And that's why I introduced Melissa now at the series 1, the Fennery moles. Of course, we all produced most data on the Fennery moles. That was the first series. And maybe it's good to share those molecules with Melissa too, if everyone agrees. We still have some in our lab here. So if everyone agrees, I will make a plate for Melissa and send it to Brazil so that she can check if any of our Fennery moles also have activity against the causative agents of sporotrichosis.
What about that DNDi azole that they're in trials with for mycetoma? Phosphoravconazole.
Yeah, that's you can test that too, but then we should test raviconazole because phosphoravconazole is produced is the prodrug, and raviconazole is the active compound. But we can easily ask DNDi if they can ship some of that to her, because officially the compound I have belongs to ESI and is bound to an MTA, so that's I'm officially not allowed to share. But we can ask if either ESI or DNDi can share.
Yeah, it would be great. Wendy, I was talking with, what is her name, that were with us in the DNDi meeting.
Fabiana.
From Brazil. Fabiana, yes, regarding the phosphoravconazole, and she told me that she was talking with them to share some of the compound to our preclinical tests here. It would be really nice to do it here. We can do with Sporotrix brasiliensis, that is the main problem here in Latin America, but also with Sporotrix schenki, that it's worldwide distribution, and with Sporotrix globosa, that it's mainly associated with Asian problems. We have all of them here. It would be nice to test.
All right, that all sounds good. Is there anything that we need to do other than, like, Wendy, you'll send those compounds? Does anyone else need to send anything through for the time being?
The only thing I guess I'd say is, I mean, it's fantastic that you're able to make a plate up of existing samples, Wendy, because that makes everything a lot simpler. If you make that up, if you could just share what it is that's been sent so we can keep track, that would be awesome.
Yeah, I will make an issue on GitHub where I also show which ones, because the ones from DNDi, I need to get approval from DNDi first, if we can share those two.
That all sounds good, but that also brings us nicely to the the point in the agenda about having a diagram with the structures of things that have been tested. We talked about that last time, didn't we? I don't know where we got to with that.
The idea was that new compounds entered into the master list should be highlighted.
Yes.
And then once they've been included in some kind of SAR diagram somewhere, that they can be unhighlighted.
Did we write that down anywhere other than in our minutes from last time?
Yeah, I don't think so. No, just in the minutes.
Okay, I'll try and make an issue or something to put that down.
So just on that, the latest data, Wendy, that you put in the master list, was that highlighted in some way that we can then just extract it out?
No, I didn't highlight it yet because there was still a lot of colors in the spreadsheet, but I can highlight it quite easily because I do know which ones I put in just a few days ago.
Right.
So those
Because-
colors that are currently in the spreadsheet, like I've got it open and I can see a couple in blue. That's the main color I'm seeing. There's some in where the text is in red. Does somebody know what all of these colors mean, or has everyone just been using different colors to mean different things?
No, the green ones are the median values of the IC50 and IC90. So those are highlighted in green, and I think
then
one
the
is in
orange.
orange.
Yeah.
Yeah, I didn't do the orange one, but yeah, for
But-
me, in our own spreadsheet, they're also highlighted in dark green.
Okay. Yeah, just having a look, like just, opening it and scrolling through. There are a bunch where the IDs are highlighted and some where the number, like the DMI is highlighted in red. Matt, do you know what those highlights are about? It's like looks like random
No.
compounds to me.
Yeah, no, I don't know what the highlighting means. I think maybe in some cases over the years people have highlighted things just to remember things and no. Yeah, so I'm not sure.
I can, if everyone agrees, I can make a black colored, so that all the text is at least black colored. We keep the green ones for the IC50 and IC, the IC50, IC90, because that's where the selection is based on, so that everyone can see those columns quite easily. And then I will make the new ones blue.
Yep, blue for new. That sounds good.
Yeah.
Okay.
Then I will I will update the table.
Okay, excellent. Do we have anyone here to talk about the modeling these pheneramyl compounds?
Doesn't
I'm not
look like
seeing
it.
any of their names. Okay, then I guess we'll move on. Wendy, you already told us last time about the fluorescence generally. So have you had a chance to do more testing? Got any new things to share?
So what we did it also in the cultured fungus. It didn't work, so maybe we should take the cytophores for now out of the discussion and go back to it later on when we have new details or new results, because we're trying to get some cytophores from the culture supernatant to characterize them. But so far we weren't successful. So maybe it's easier just to get it out instead of going back to it every time.
Yeah, that sounds fair enough. Alrighty. I don't think we have anything else to talk about to do with pheneramyls specifically then. So if we jump to series 2, I will start off by saying I have not yet organized another schools meeting, but I will do that very soon. So I will do it a similar way. I'll make an issue on now. Actually, there are some of you, some people are here from the schools. Previously, I was making issues in the aminothiazoles series 2 repository, but given that some people are making phenothiazines and other things now, is it more helpful to put them in this repository, the monthly Zoom meeting one?
Yeah, maybe. If it's going to genuinely be across series, then maybe, yeah.
Yeah, because
sure.
previously we were just the one, but yeah, that's okay. So I'll put that. I'll try and have that. Try and have something up about planning a meeting by the end of the week. I'm not sure when the meeting will end up being. And I don't think any more testing has been done since. Well, I should just ask Ma. Has any more testing of those compounds that we've sent been done? Or are we up to date there? So that's the aminothiazole
The initial screenings have been done, I think, and updated in the list. Marij is currently, I think, working mainly on the MIC50, so against different fungal strains.
Okay, excellent. And is that that would be compounds that have been shipped from us at UCID and then from the Uni of York and UCL?
Yeah, so they're from different sources, and we all screened them in one big batch, and those results are on GitHub. So I updated where we were, well, just a few days ago. So all the updated results are in the spreadsheet. So everyone should take a look at the spreadsheet to look for their own compounds, and I will highlight the ones I put in last week.
Yes, okay, that sounds great. I will put any that are from UCID on our SAR giant diagram. If anyone else would like me to put compounds from their uni, like to update their ones, let me know. But otherwise, I'll just stick to the UCID ones for now.
Sorry, just a quick question. Wendy, do you know whether there are going to be any updates, further updates on the IC50 values for the latest cytokines that we sent to you? Those 5 compounds. You updated the spread. You and Ma updated the spreadsheet, and there were percentage growth, fungal growth percentages only at 25 micromolar and 100. But I think 3 out of 5 compounds gave full inhibition, which means we usually get like the IC50s and minimum inhibitor concentrations. Do you still plan on updating that, or is that going to be final?
Everything which has an hit at 25 micromolar is currently screened or has been screened. So I have to double check if those results are not there yet, if they are still ongoing. Marij, I think you are here.
Or I can send you a message just to speed up things.
Yeah.
With specific numbers.
Yeah, that's easier.
Okay.
But we are still screening, and everything which was not put in probably will be put in quite shortly. But the in vitro work is still ongoing.
Okay.
I don't know if anyone has heard from Seven Oaks lately about compounds. I certainly haven't. But I can see that Peter Rayner is here. I don't know if you have anything to tell us about what compounds you're going to be making in the future or what you've already made.
Yeah, so our students are back in the lab. So we updated the issue with what we plan to make, and that is still what we plan to make. There's a couple of other ones that since I will update with, but yeah, I think that they're where we're at. And then we probably have some students starting in the not too distant future who might work on series 3 type compounds. So just simple ones, but yeah. So yeah, we're progressing.
Okay.
We'll have another batch to send off probably in the next few weeks.
Great. I will probably ask you more about this later in our schools meeting, but are you going to be making phenothiazines for the foreseeable future now? Is that the plan? Or
we can do whatever, really. We're not wed to anything.
Okay.
Whatever is best overall. But judging by some of the recent data that came back last couple of days, there looks to be some interesting compounds that we could make. But yeah.
Okay, cool.
Can I ask a question? Peter, what's just remind me, what's the level of the students and how long do they have in the lab?
So we have year 3 BSc students in the lab who've been in there for probably 12 weeks now. And then we have a smaller project, which is year 3 MChem students who are in for 8 days. So the BSc students have made between them probably 40 compounds. The MChem students might make 15 or so, that kind of number. But yeah.
Okay. That's awesome.
Yeah, that's good.
Sorry, endless questions. Are the students keeping lab notebooks that can be shared?
Yes, they are, to varying standards, I would say, but yes, they are.
But
so
are you able to, link to them and share publicly kind of thing?
Yeah, we will do. Yeah. So a lot of them, I think we put them somewhere. I will find the links, but I can also upload all their final reports once they're done as well, with everything in. But yes, yeah, we can do that.
So I mean, the links to the raw lab notebooks are awesome. If they ever go offline, then that kind of backup copy somewhere is also good, because sometimes these shorter projects they use them for a bit and then stop.
Yeah.
Also, just incidentally, are you on the invite for these meetings?
I am now. Yes. Yep. After last time. Yep. So no, that's good now.
Awesome. Fantastic.
That's great. Lovely.
Sorry, just on that as well. What are they using to keep their lab notebooks?
They are using lab archives. Yeah,
okay.
At the moment. We can use whatever though is we told them to use lab archives for this one, but yes, whatever they want to, or you want us to, is fine.
Yeah, I think lab archives has been used successfully for a while now.
It is unusually easy to share openly versus lots of other lab notebooks.
Yeah. So that's good.
I'm guessing that Andrea is not joining us today, given that it's like 2 a.m. in New Zealand. So we should probably, don't think we have anything new from her on benzimidazoles. Had she sent a batch to be tested recently? Sorry, it's a bit, I'm struggling to tell from what we have in the agenda. But Wendy, Ma, have you been testing any benzimidazoles, or is that all testing that has been done in the past?
No, I just updated the NEU compounds, and I think the other ones were from the OU compounds. They have been added a month ago or so.
Okay, so I guess we'll just wait until Andrea can join us, which hopefully will be next month when it's a more friendly time. And they're going to be making benzoxazoles in the future. Okay. I have not yet been able to get Marita to put her raw data and so forth online, but she is back. I've seen her recently, so I should be able to sort that out soon. Okay. Then we have ketoxins again. Dimitri, you're probably the person who has the most to say about this at the moment.
I think I shared the updated scheme straight after the last meeting, so that has been sorted. And we discussed it last time, because I can see in the minutes from the previous meeting that there is an update about the oxyconazole nitrate, which was the salt formulation, which gave so far the best result in the series.
And just we have like a note about putting things under future plans there, and looking at those different salts.
You mean the wiki? Yeah.
Has somebody done that yet, or would somebody like to volunteer to just quickly do that at some point soon?
Are we talking about the ketoxime?
Just that we have this note that I remember has been here for a while about how, we have the suggestion of testing different salts, but presumably why is that as a future plan?
So I will update this when I finish my dissertation, because I'm still getting some conclusions on that anyway.
Okay, so you're going to have some more things to put on
the
there.
work
Okay.
anyway, yeah.
Do we still need to keep this sentence in our agenda though, or can we remove it for now?
I think we're okay to remove it because I'll still be updating before I finish the PhD.
Cool. And we've already talked about our phenothiazines and the global health priority box. Ma tested that previously. Some were being taken to in vivo. We have this molecule MMV 1804559, and there's some discussion that I haven't had a chance to look at, but
I managed to do a little bit of data mining on it, and it's got a bunch of different names. And there's another problem, which is that the stereochemistry is defined in some, but not in others. So we don't know exactly. Let me just try and pull that up just to, I think you should have all seen this by now, but
Can we buy it?
I don't believe so.
Can we buy anything that's similar to it?
I'm not exactly sure, but I do believe that pharmacophore is common in kinase inhibitors. So I think this is actually probably some sort of kinase inhibitor type drug. But you can see, hang on, let me just try and get this to one moment.
I just think it's a significant result that Wendy's found this thing. I mean, the stereochemistry is likely to be, it's just to the right of where your circle is. The stereochemistry is not defined, but it's likely to be racemic, and it probably doesn't matter too much.
Yeah, I was about to figure that, but some of these entries are only for particular isomers, but this has about 3 different names. So Chembel 5277240, TCMDC. But I don't see these for purchase, but I assume it was probably part of a kinase library.
Yeah, possibly. If it's TCMDC, then it's a molecule that probably ultimately came from GlaxoSmithKline.
Yeah, that's what MMV told me, that it's from a pharmaceutical company which didn't use it anymore. So that's why it ended up in these open boxes. We do have some hunches where it might work. There's in fungi, there's a molecule, or it hits a target probably associated with the Warren body. So I did a search for homolog in Madurella mycetomatis. I found a homolog. The only thing is maybe we should model the molecule inside this homolog, but I can't do that. So maybe there's somebody who can to see if that's really potentially the target.
Of that molecule?
Yeah. Well, we did some searches in PubMed, etc.
If it did that, would you expect it to have a change, to make a change in the morphology of the grain?
Well, it's a rescue mechanism of the fungus, so that in knockouts of that particular gene, they found with other fungus also a small lesion, so that it decreased the lesion size of the fungus itself. So it has a similar phenomenon as we note here in our larvae. So in that respect, it does what it does in other fungi too. So that would make it an ideal target because the Warren body is fungal specific and Ascomycete specific actually. So then you wouldn't expect too much toxicity issues maybe if you fine tune it towards the fungal variant a little bit more.
I mean, so, the quick, rather than the modeling, a quick thing to do would be to, an experimental assay if we could find the compound and analogs of the compound and evaluate them in some way. Is there such an assay, or is it actually your assay the easiest one?
Yeah, you cannot do this. We thought of it too, but we can't do this quite easily. We cannot make knockouts or whatsoever in Madurella mycetomatis. So it's not like we can assess it against the knockout. That's what you would normally do to determine if it was indeed a mode of action.
I think I need to do some reading on the Warren body. It's new to me.
It's actually a plug. So if you have a fungal hyphae, it's not a cell adjacent to a cell, but it has a pore in between. So the risk of when a fungal cell is damaged, if you wouldn't have the Warren body, it would lose all its cytoplasma in one go, because all the different cells are linked to each other because of these pores. And that's why they have this plug called the Warnin body, which positions itself inside this pore so that it would not leak out all its cytoplasma when one of the cells is injured.
Right.
But it's not the Warnin. It's not the Warnin body itself. It's one of the enzymes positioning the Warnin body inside the pocket.
Right, that's the target I'm thinking of. Yeah. So if there's an assay for that, then that's the simplest thing we could do.
Yeah, but I looked in literature. There's actually not.
There is very little data on the molecular mechanisms of this Warnin body. I'm just doing a PubMed search, and there's not much.
No, it's an ideal target for an antifungal compound, but not much is known.
Interesting. I think it's a really significant compound, though, and we should keep it on the on the kind of dashboard, because I don't know how common it is for you to have seen compounds that do modify the grain morphology, but those grains are one of the
not
biggest
like this
challenges we have. Yeah, it's good. It's an interesting compound.
Do we have anyone at the moment who can make some analogs on this compound, or is everyone, all of our chemists, busy writing PhDs and things at the moment?
Yeah, or maybe someone who can do a search where we can find some, because that's not my expertise. So if there are analogs which somebody already made, then we could ask if they could share.
Yeah,
so I might be able to do a search on the pharmacophore. I can try. It looks like a very common kinase inhibitor type thing, so I'm surprised none of the others kinase inhibitor boxes hit.
Yeah, so I think A, can we buy this? B, can we buy things that are similar? And C, if that is a GSK compound because it's Trescantos, then we could reach out to that team. I'm happy to do that and see if they have anything related to it. Those would be the first things. I mean, before we do any chemistry, because I mean, it's not a trivial compound that. Before we do any chemistry, we should see what's available.
So did we establish that we cannot buy it? Is that?
Sounds like it. I haven't searched.
Yeah.
I asked MMV, of course, and they said that it's a private compound. It's not a commercial compound.
Right. Yeah, when you do the search on the code, you don't get very much. So the implication is that it's come from the library, but there must be more of these. So I'm happy to reach out to GSK.
Yeah. Okay. That sounds like the most.
Yeah, that's probably
likely to be helpful.
This is actually pretty. This is a pretty complicated molecule.
Yeah.
And then on to more kinase inhibitors. There is more data, more drugs from Bernhard that's screened by Mariah. That data has already been reported and uploaded, but not yet summarized, judging from this agenda. Who is it that that would be summarizing that or could be summarizing that?
I think Mickey Konings is on the call too. It's part of his PhD project, so probably he's writing it. He should write it up. Mickey, do you want to make a comment?
Sure. Let me add the camera so that you can see me as well. It's not really that interesting at the moment. Or the thing I have to share is not that interesting because I'm in the process of writing it up, but I'm also doing some other thing or finalizing some other things at the end of my PhD. So this is on the to-do list, so to say. So hopefully during one of the next meetings, I'll be able to give a better update.
Okay, sounds good. Bioinformatics, Walid was going to be generating a phylogenetic tree. I don't think anybody talked about this last meeting, and I don't think we've got anyone here to talk about it today. Okay. The plant extracts from Nigeria. So firstly, have those extracts been evaluated? I think there was like a labeling issue previously. Is that right, Wendy?
Yeah, so what we asked last time is that someone could share how they look like, because we cannot locate them at the moment in the lab. So that we can easily find them and by which temperature they were supposed to be stored. Because, well, they are not in the drawer where we keep all of the rest of the compounds, and also not in the -20 where we keep all of the other compounds. So we don't know where they exactly are and how they look like. So that makes it difficult to search.
And I'm assuming
so,
you didn't hear back from anyone when we talked
I
about this
will
last
ask
month.
Dr. Ogachi again if he can provide information about the labeling. The problem is these are plant extracts, so it's not easy to add it to the list because they're not pure compounds like the other compounds on the list. But yes, I will get back to Dr. Ogachi and ask him. And well, if they are not available anymore, or not, if you can't find them in your lab anymore, then maybe you can send them again, because plant extract preparation is not too complicated and should be easily done within a short time.
Yeah,
Then the shipping.
It
The
would
shipping
already
to
help
ask.
quite a lot what kind of vial we are looking for.
Ah, okay. Yes, of course, yes.
Can I just
And the question
ask
then,
why, like, what's like the backstory to these compounds in 5 seconds? Like, why are we testing?
well, they are extracts of medicinal plants from Nigeria, from various regions of Nigeria. It's a quite big country. So, and then they're using it for infectious diseases, for treatment by local healers, for example. And they are preparing extracts and testing them for their own issues they have. And of course, I asked them if they can send some samples for testing against mycetoma. And yes, that's the history behind.
This was from the cinnamon oil research, right? The CIN compound that we found that was antifungal, right?
Yeah, it can be. I exactly don't know what exactly they are consisting of, these extracts, but of course I can ask them again about them, more infos about it. And well, if it's necessary to send them again, then I think it can be done easily. Maybe paid by DNDi again. I have to ask Luisa about it anyway, because Dr. Nuisi has some new samples from southern Nigeria, plant extracts from southern Nigeria now, and he was already asking if he can ship them now to Wendy's lab. And yes, I will care for it.
Yeah, if we can get them all in one go, that would be simple. And yeah, you already mentioned that it's difficult to assign codes to those samples. Is the plan at the moment to just have a description in our master list, just not worry about assigning a specific
Exactly, I
number?
don't have, so because he sent it directly to Wendy's lab, and I don't know their own label from their own lab, the lab numbers or lab labeling. But I will ask him again how he has done it.
Even just a list of species that the oils were pulled from is good.
Would be also fine to know, yes.
Yeah, and maybe
we can have an ID. There's no problem in assigning a code to it, because that just means we can refer to it and put something in the spreadsheet. But there needs to be a note somewhere that says that it's not actually a purified molecule; it's just an extract. And that way, you're allowed to have then batches. So you could have, if it's the same extract, then you've got the same code, and you just got a different batch number if you prepare another sample of it. That's okay. It would help with tracking, that's for sure, because there's no record online of these samples or what was sent, so it's difficult to track.
Yeah, so if we give them a MycetOS number, we can just that's already got an inbuilt batch number. And yeah, I mean, I suppose where we normally put in the details about molecular weight, somewhere there you can just write a note that we don't know the molecular weight.
Yeah, because there are mixtures of compounds, you don't have exact molecular weight or SMICE code, so you must handle them a little differently, but I think that can be managed.
Yeah. Okay, on to our miscellaneous ones. Matt, I think you had to leave before you could tell us about the DMPK measurements last time. So, or you maybe hadn't had a look at them yet. I can't remember.
No, so we got them, and the issue is that the spreadsheet that we got, which has really useful data in, has data from lots of different people. And so it includes data for compounds for mycetos, but from several series. So it includes phenaramols, ketoximes, and I think there's some aminothiazoles maybe. And also data from other projects, right, that were measured at Dundee. And so what I couldn't just dump the spreadsheet onto GitHub because it has data from other projects, and I don't want to take responsibility for those. So we just needed to delete out the projects, the data for compounds which are not ours, and then summarize in a diagram the data for the compounds that belong to mycetos. That's all. And I thought we'd arranged that, Dimitri, you were going to do that because some of the data were for compounds that are relevant to you, to extract the data and diagrammatically present them. But I think that's where we got up to. I couldn't share the original sheet because it wasn't completely ours.
Okay. So then,
I don't remember we had this conversation before, so it's the first time I'm hearing this.
Okay, so in that case,
I'll
I'll take the sheet and I'll separate out the data for mycetos and then generate a new sheet. And then people from mycetos can take a look at the data and extract the data that's relevant to them. That would be the best thing to go for.
so we each make a summary of the compounds that we've taken responsibility for.
Yeah, and it's very useful data. And it's good to compare them all together because you kind of see what the range is, but it's really useful to have. I mean, it's great that they did those measurements for us.
Yeah, okay, that sounds good. I have once again not yet given Rowan Davies compounds myos codes, but I will definitely get it done tomorrow. I'll prioritize that. And then we just have, I think, again, something we've discussed before, a comment about the toxicity studies that Ma has shared with us previously, but how we put that data into our master list and into our wikis and summaries and so forth. Have we come up with any better strategies on how to do that?
What we could do is we add a column simply in the master list that toxicity testing, toxic, yes, no. It's a binary thing because it's based on if the survival curve differs from that of the healthy control larvae treated with the solvent, and statistically, or if the survival is below 80%. So that's basically why we say, yes, it has toxicity issues, yes or no. And if we test a different concentration, or maybe we say toxic, not toxic at, and then we simply state the concentration where it's not toxic. Maybe that's the easiest. Because for some compounds, we were able to test at 10 micromolar instead of 20, because 20 was toxic and 10 was not. So maybe that's then the issue. And we leave the ones which we didn't test blank.
Yeah, so that's just an extra column in the master list,
Yeah.
and it has either a number or if it hasn't been tested, it's left blank. That's
yeah.
That sounds like a good place to start. Let's see. Yeah, I can't see anything else there that, you know, we've said, is there a good way to share existing other data like ADME data, etc. But we don't really want to put all of that in the master list, I'm assuming. Or do we? Is there?
Sorry, for which data are we talking about? Sorry,
I'm looking at my own summary from whenever this meeting was, September. So at the moment, we collect data on larvae toxicity, which is what we were just saying. We can put a column in the master list for that. But some of the compounds have data for other things like toxicity data, etc., but not the larvae specifically. I think this was specifically the compounds from Laurie Ferrens. Apparently she has lots of data that is not, there's nowhere to put it in our master list at the moment, but obviously we'd like to have it.
Those were for some of the tricyclics she her group had made. They were not phenothiazine, but they were tricyclics that were somewhat similar in structure to chlorpromazine. So we got them screened, and we did get results. We did get 2 compounds that hit. One was a little bit, like, it was quite large. It had 2 Bach groups on it, so it might not be the best, but the other one was kind of more similar to something like chlorpromazine or promethazine. Or so that was, I posted the structure of the hit in the repo for phenothiazine tricyclics. So Peter, you should have already, Dr. Rainer, you should have already seen that, right?
Yeah, we've seen that. Yeah.
Yeah.
So yeah,
it's kind of similar. It's got a carbazole instead of a phenothiazine, but you can see they're all kind of sort of structurally in the same ballpark.
Yeah.
And so those molecules, Laurie already had a bunch of data on, and has she given us that data? I feel like I remember her sharing links with us somewhere, but did we put those links on a wiki or anything, or do we need to?
Yeah, if it's there, if we have it, then if it's a certain assay and a number for a bunch of compounds, then we should try and add that to the master list in a new column.
Okay,
but
so as
that
a new
means
column.
it means being specific about what that is, because not all toxicity measurements are the same. So we'd have to put in a column which says toxicity measured at, and then wherever it was, AstraZeneca or whatever it was, I forget who did the assay, and then put the data in for those compounds only. And if someone else measured toxicity with another assay, it would need to be a different column. The alternative to that, if that's messy, is to have a tox column, and then we dump the sheet somewhere which has the data in it, and then we get the link for that sheet, and we just dump that into the relevant cells in the toxicity column, so that if you want to know what the toxicity is, you can go and find it. That then makes the spreadsheet less useful, because they're not numbered, so you can't just sort them. But it's a quicker fix if you want to do that. I don't know what the best way of doing that is. Ideally, you put the numbers in, but it means someone's got to extract them and actually add them.
Yeah, I don't know either. Would it be ridiculous to have another spreadsheet that was specifically for toxicity, so that it was a little bit more manageable, or is there too much data that we'd be copying across in like anyway?
Yeah, it's spreadsheet divergence. You just got to
don't want that.
I don't know. I think that if it's just a bunch of numbers for a bunch of MycetOS codes, then we should try and add it in. But the data that she sent does need to be somewhere, right? The sheet needs to be somewhere that she sent us, and then we can link to that no matter what. So the first thing I think would be to make sure that we've got the tox data for those compounds somewhere.
Yeah. Does anyone remember how she sent it to us, where it was sent, what issue, etc.? I suppose we can probably go back through that September meeting and see if we have any notes on it, but I'm not sure that we do.
All right, action item, find it.
Yes, definitely. Okay. Otherwise, I think these notes on screening methods we have already covered previously. We've talked about making sure everything has very clear MycetOS codes, or at least institutional codes, etc., if they're being sent to Erasmus. We are trialing our color code system. This says that the biology team is going to highlight the samples in yellow, but we've said we'll do it in blue instead. And we'll remove that blue color once a chemist, someone, has had a chance to put that data into a summary somewhere. And then we just talked about tox data already. Okay. Was there anything else anyone had to mention before we wrap up? Okay. Well, looks like our next meeting is March 26th, and it's 8 a.m. for you guys in the UK. I'm not sure what time zones there are for other people. And yeah, I guess that's all we have today.
Yeah, remember it's a difference in the time because of daylight saving. So you probably go the other way around than we are.
Yes, I actually have no idea when daylight savings changes in Australia, but I will certainly check.
Yeah, that was also one for the minutes, so that everyone should check with the daylight savings.
I've got the link there, I think, for the.
Yes, yes, you do.
You should have it in there in the link.
Will it have it for the daylight savings? Oh, it does. Oh, that's excellent.
Yeah.
Okay. That's really helpful.
Right. All right. Thanks for sharing. Great.
Alrighty. See everyone in a month.
Yep. Take it
Bye,
easy.
See you
everyone.
later. Bye.
Bye, everybody.
Bye.